Hello everyone. In this class, we'll try to learn about what is synaptic transmission. The objectives of this class would be to give the classification of synapses, draw a label diagram of synapse, describe the mechanism of synaptic transmission, list the properties of synapse, and give examples and mechanisms for different types of synaptic inhibitions. Communication, the major function of the nervous system, is achieved through synaptic transmission, the transmission of impulses through synapses. The synapses are the neuronal junctions through which information from one neuron passes to other. Though the synapses, the neuronal messages are conveyed to the appropriate target structures in the CNS. The command signals from CNS to the peripheral organs are also conveyed through the synapses. Higher functions of CNS, that is processing, integration, information, learning, memory, are also possible because of the activation and alteration of activities at synapses. Formation of new synapses and synaptic modification continue throughout the life of an individual. There are different types of synapses. There are innumerable synapses in the nervous system. On an average, the neuron forms about 2000 synapses. As there are about 10 raised to 11 neurons in CNS, the total number of synapses may be about 2 into 10 raised to 14. These synapses are of different types and have various names. Synapses are usually classified as follows according to the part of the neurons involved in the formation of synapse. Based on the parts of the neurons involved in the formation of synapse, four types of synapses are found in CNS. These are called as oxodendritic, oxosomatic, exoexonic, and dendrodendritic. Exodendritic synapse. In this type of synapse, axon of presynaptic neuron connects with the dendrite of postsynaptic neuron. This is commonest type of synapse in central nervous system. The axon terminal may synapse with the spinous process of the dendrite, so that is why axospinous synapse, or with the shaft of the dendrite, where it is called as shaft synapse. Exosomatic synapse. In this variety, the axon of the presynaptic neuron synapses with the cell body of the postsynaptic neuron. So this is also a common form of synapse in the central nervous system. Exoexonic. In the exoexonic type, the axon of presynaptic neuron synapses with the axon of postsynaptic neuron. In dendrodendritic synapse, in this type, the dendrite of presynaptic neuron connects to dendrite of postsynaptic neuron. In CNS, axodendritic synapse is common type of synapse followed by axosomatic type. For example, in cerebral cortex, about 80% of the neurons synapse on dendrites, whereas only 15% end on cell bodies. Exoexonic synapse is less common and dendrodendritic synapse is rare one. The ratio of synapses to neuron in human forebrain is about 40,000 is to 1. So these are the different types of neurons. The exospinous type of synapse, shaft synapse. Okay. So then axon ending on the body and where the axon ending on the presynaptic membrane. According to the nature of transmission, synapses are of three types according to the mode of transmission. It can be a chemical synapse, it can be electrical synapse, or conjoined. Chemical synapse is a transmission of impulse occurs through the release of neurotransmitter, whereas in electrical synapse, transmission of impulse occurs through the gap junctions. And conjoined types, they are partially electrical and chemical. According to the number of neurons involved, the one neuron ends on another, that is 1 to 1. Multiple neurons terminate on a single neuron's many to 1. The one neuron contacts multiple neurons, that is 1 to many. Example of 1 to 1 synapse is the neuromuscular junction. Many to 1, this is a usual type found in CNS, and 1 to many is less frequent. In autonomic nervous system, usually one-to-one synapse is found in the parasympathetic and one-to-many type is found in the sympathetic system. According to the nature of transmission, synapses are categorized into chemical and electrical synapses. As chemical synapses are very common, the electrical synapses are sparse in CNS. The mechanism of synaptic transmission is discussed under this section through the chemical synapse. The functional anatomy of a synapse. The neuron from which the information passes through the synapse is presynaptic neuron, and the neuron which receives information is the postsynaptic neuron. The part of presynaptic neuron axon terminal forming the synapse is called as presynaptic membrane, and that of postsynaptic neuron is called as postsynaptic membrane. The space between presynaptic and postsynaptic membrane is called as synaptic cleft. The presynaptic axon terminal, postsynaptic membrane, and synaptic cleft form the synapse. Presynaptic axon terminal. The presynaptic membrane is the part of axon terminal of the presynaptic neuron. The terminal of the presynaptic neuron typically ends in small bulbous enlargement called terminal button or synaptic knob, which is about 1 mm in diameter. The synaptic button contains two specialized structures, that is the numerous synaptic vesicles that contain neurotransmitter and dense stuff or projections that made up of filamentous proteins which contact larger filaments in the exoplasm. Synaptic vesicles, there are three types: small vesicles with a clear core, small vesicles with a dense core, and large vesicles with dense core. The small clear vesicles mainly contains acetylcholine, GABA, glycine, or glutamate. Small dense core vesicles contain catecholamines, and large dense vesicles contains mainly the neuropeptides. Large vesicles are present throughout the presynaptic terminals and are released by exocytosis from all parts of the presynaptic membrane, whereas small vesicles are located close to the presynaptic membrane and are released into the synaptic cleft through the active zone by exocytosis. The small vesicles, after discharging their content, are recycled back into the terminal. The vesicles are endocytosized, then fused with the endosomes. New vesicles that are formed from endosomes by budding are refilled with the transmitter chemicals. Vesicles then move close to the active zone and undergo docking and priming before discharging their content into the synaptic cleft, the next cycle. Dense stuffs are filamentous projection present in close in contact with vesicles and play an important role in exocytosis of vesicles. Dense stuff are present mainly in the active zone. The presynaptic terminal also contains few large mitochondria. A region of a presynaptic membrane is modified to form active zone which contains many proteins and calcium channels. The release of vesicles containing neurotransmitters occurs mainly through the active zones. The synaptic cleft is the gap between pre and postsynaptic membranes, which is about 20 to 50 nanometers wide, and this space is filled with extracellular fluid. Transmitter molecules released from presynaptic terminal diffuse across the cleft to reach the postsynaptic receptors. The postsynaptic membrane usually is the part of the dendritic spine, but may be a part of cell body or a part of axon which contains receptors for the neurotransmitters. The area of postsynaptic membrane modified for synaptic transmission is called as postsynaptic density. The postsynaptic density is the cluster of receptors for transmitter embedded within the postsynaptic membrane. Neurotransmitter is the chemical substance used for transfer of information through the synapse. The neurotransmitter amplifies the effect of action potential arriving at the synapse. Due to this amplification, presynaptic action potential after passing through the synaptic cleft, stimulates the postsynaptic neuron. The arrangement in the synapse is such that the neurotransmitter released from the presynaptic nerve ending acts on a wide area of postsynaptic membrane and activates large number of receptors, that is ion channels, to activate the postsynaptic potential. So this is the diagram showing the synapse, which consists of presynaptic neuron and the postsynaptic neuron. And in the presynaptic neuron, you can see multiple mitochondria, the vesicles, which can be of small or larger vesicles. You can see the dense stuff and the active zone. Okay, and where in the postsynaptic membrane, you can see the postsynaptic density, and you can see the ion channels and the receptor complex. Okay. And the middle zone is called as the synaptic cleft, that is the space between the presynaptic neuron and postsynaptic neuron, where the neurotransmitters are released. And there can be a recycling of neurotransmitter which occurs in 6 steps. Okay, so the first, there will be discharge of the contents by the clathrin coated vesicles. So then the clathrin molecules, they get separated and they join early endosomes. Okay, so then there will be formation of the early endosomes. Okay. So then there can be a process of docking and priming. Okay. So these docked and primed vesicles, they are again further going to release the neurotransmitter into the synaptic cleft by the clathrin mediated exocytosis. Neurexins are the proteins attached to the presynaptic membrane. There are neurexin receptors in the postsynaptic membrane to which neurexins bind. Thus, neurexins that link presynaptic and postsynaptic membranes provide structural stability and synaptic architecture. In fact, the systemic organization of the synapse depends partly on the neurexins. In many vertebrates, neurexins are produced by a single gene that codes for alpha isoform. The neurexins are coded by 3 genes, and there are both alpha and beta isoforms. As each of the genes has 2 regulatory regions and extensive alternative splicing of their mRNA occurs, so there are more than 1000 types of neurexins. It is proposed that neurexins not only bind to the synapses together, but also they provide basis for synaptic specificity. Steps involved in synaptic transmission. The synaptic transmission is the process by which the information from presynaptic neuron passes to the postsynaptic neuron through the synapse. In a chemical synapse, it occurs due to the release of neurotransmitter from presynaptic nerve terminal that initiates action potential in the postsynaptic neuron. The mechanism of synaptic transmission can be divided into presynaptic and postsynaptic mechanisms. Presynaptic mechanisms, there are 5 major steps involved. The first one is vesicle containing neurotransmitter molecule that are concentrated at active zones of the presynaptic axon terminal and undergo docking and priming. Docking is the process by which vesicles attach with a membrane, and priming is the process by which vesicles become ready to discharge their content in response to a stimulus. The action potential that arrives at presynaptic axon terminal depolarizes the presynaptic membrane. The depolarization of membrane causes opening of voltage-gated calcium channels that allows calcium to enter the axon terminal through the active zone. Increase in calcium concentration in the presynaptic terminal increases the calcium-mediated exocytosis of the vesicles. The calcium causes fusion of vesicles to the presynaptic membrane by causing contraction of the microfilaments in the dense stuff that facilitates their movement and then help to discharge their contents into the cleft. The discharge of synaptic vesicular contents takes place through the small hole in the cell membrane, which immediately closes rapidly. In this process, the main vesicle remains inside the cell, and this is called as kiss and run discharge. Following discharge, some vesicles quickly recovered by endocytosis and refilled locally, in which the endocytic process is short-circuited. The transmitter is released into the synaptic cleft in a quantized amount that diffuses passively across the cleft to the postsynaptic membrane. The quantal release of neurotransmitter is sometimes called as Dale's phenomenon. From the calcium influx to the neurotransmitter release, it takes about 200 microseconds. Normally, the small synaptic vesicles recycle in the presynaptic nerve terminal. The mature vesicle move to the active zone, dock and get primed. When the action potential arises, the calcium influx facilitates the fusion of vesicles with presynaptic membrane that causes discharge of granular content into the synaptic cleft. The vesicle membrane then gets coated with clathrin and taken up by endocytosis into the presynaptic terminal, where it is reutilized for the neurotransmitter packaging. Fusion of synaptic vesicle with cell membrane is facilitated by synaptobrevin, a V-snare protein present in the vesicular membrane, and syntaxin, T-snare protein present in the cell membrane. In fact, synaptobrevin attaches and interacts with the syntaxin for docking and priming of vesicles. Various other synaptic proteins, SNAP25, connected with syntaxin, and alpha and gamma SNAPs are connected with synaptobrevin, facilitates the interaction between synaptobrevin and syntaxin. A multiprotein complex regulated by small GTPase like RAB3 also participate in the process. Many neurotoxins inhibit the neurotransmitters by preventing attachment of synaptobrevin and syntaxin. For example, tetanus and botulism toxin act on the synaptobrevin and syntaxin that in turn prevent fusion of vesicles with membrane that blocks the release of neurotransmitters from presynaptic terminals. Botulism toxin are of 7 types. A and B botulinum toxin, they act on the SNAP25 and prevent interaction between synaptobrevin and syntaxin that in turn prevents docking and priming. Botulinum C toxin acts on the syntaxin and prevents its attachments with synaptobrevin. Botulinum toxin B, D, and G act on synaptobrevin and prevents its attachment with the syntaxin. Thus, botulism toxin produce flaccid paralysis by inhibiting the release of acetylcholine at the neuromuscular junction. Tetanus toxin acts on synaptobrevin and prevents its attachments with syntaxin. Tetanus toxin prevents release of other neurotransmitters from the vesicles at different synapses in CNS and therefore produces spastic paralysis. So this is the diagram showing, how the vesicles, how they are going to be docked and primed with the interaction of the synaptobrevin and the syntaxin. Okay. So when there is an interaction with other proteins like SNAP25 and other SNAP of alpha and gamma, so there will be the process of the docking and priming. So this is another general outline how the synaptic activity occurs when action potential arises at the presynaptic membrane. So it leads to the opening of the voltage gated calcium channels. So that leads to entry of calcium into the presynaptic terminal. It releases the neurotransmitter by calcium mediated exocytosis. Then binding of neurotransmitter occurs with the receptors on the postsynaptic membrane. So it leads to appropriate ion channels. There will be movement of ions through the postsynaptic membrane. So there will be change in the membrane potential in the postsynaptic membrane, which is going to lead to the generation of the synaptic potential, and so which is going to in turn transmit the impulse to the next neuron. So this is in brief about the synaptic activity. Thank you.